Hello friends, so today's topic is mitochondria. Mitochondria introduction. Mitochondria is the powerhouse of the cell which produces energy. It is a membrane-bound organelle present in the cytoplasm of the cell of eukaryotic organisms which synthesizes energy molecules in the form of ATP, which is used by the cell. Hypothetically, mitochondria are believed to have originated as prokaryotic cells like bacteria. They were capable of oxidative mechanisms. Over the course of evolution, they started living as endosymbionts inside the cell of prokaryotes. Mitochondria have fragments of genetic information in the form of DNA and can multiply on their own by division. What is a mitochondrion? Mitochondrion is a double membrane bound organelle which is found in the cytoplasm of most of the cells of most eukaryotic organisms. This little center for energy generation digests the nutrients and releases chemical energy in the cell in the form of ATP. ATP is adenosine triphosphate. Apart from generating ATP molecules, the mitochondria also regulate cell growth and cell death. It signals the cells and generates heat. It plays a major or vital role in cellular respiration. Anaerobic fermentation is another process by which ATP is generated in the body. But anaerobic fermentation doesn't take place in mitochondria. Mitochondria necessarily need oxygen and glucose to generate ATP. The energy is generated through this process is more than the energy generated by anaerobic fermentation. Structure of mitochondria. Mitochondria in animals are round or oval. Mitochondria in animals are round or oval and are bound by a double membrane. These membranes are composed of phospholipid bilayers and proteins. The different parts of mitochondria in animal cells are outer membrane. The outer membrane keeps the inner organelles intact and in place. It is made up of protein and phospholipid bilayer. The outer membrane contains the enzymes involved in various activities. This layer is also permeable to macromolecules so that ions ATP, ADP, etc. can easily pass through this membrane. Then intermembrane space. The space between the outer membrane and inner membrane is the intermembrane space. This space is also consists of small molecules like ions, sugars. As the outer membrane surrounding it is permeable to these molecules. Inner membrane. The inner membrane of the mitochondria is made up of proteins that carried out different functions. It also has enzymes that are necessary which catalyzes processes necessary in the production of ATP. The inner mitochondrial membrane is permeable to oxygen, carbon dioxide and water. The inner mitochondrial membrane is the fundamental site of ATP generation. The inner mitochondrial membrane has several folds in it called as cristae, to increase the surface area. Cristae. The inner membrane of mitochondria is folded into several folds. So these folds are called as cristae. The cristae increase the surface area inside the mitochondrial membrane to generate ATP molecules efficiently. The larger the surface, the more space to carry out cell functions. The surface of the cristae has exosomes attached to it, which aids the osmosis of ions. A lot of chemical reactions occur in the inner membrane on the surface of the cristae. Then matrix. The space of the inner membrane apart from the cristae is matrix. It is fluidic and contains proteins, ribosomes, enzymes, tRNA and genomic DNA. Due to the presence of genetic material, the mitochondrial matrix can synthesize its RNA and proteins. The synthesis of ATP would be incomplete without the enzymes of the matrix, which is the most important chemical cycles. So functions of mitochondria. The prime function of mitochondria is to produce energy. Okay. It is the power generation plant where the nutrients turn into ATP by chemical processes. The other major role played by mitochondria is to carry out cellular metabolism. Through cellular metabolism, three major processes are carried. First one is conversion of food into energy. Second one is conversion of food into molecules that are essential for the body like proteins, carbohydrate, etc. Third one is elimination of wastages. Mitochondria is also responsible for cell growth and multiplication. When there is too much strain on an organ performing a specific task, mitochondria multiply on their own to carry out their purpose more efficiently. It is also plays a major role in apoptosis or cellular suicide. During apoptosis, a cell doesn't die of any injury, but a series of chemical reactions leading to the elimination of the cell. It is also responsible for hematostasis by heat production. It happened by the process of mitochondrial uncoupling, wherein the re-entered protons in the matrix are not converted to ATP. It stores calcium ions, thus helping cell signaling. Free calcium regulates a series of chemical reactions in the cell and thus mitochondria regulate cell signaling. Apart from these, mitochondria regulate cell differentiation, cell senescence, or the cessation of the cell division. Facts about mitochondria. Mitochondria are colorless organelles. Therefore, they cannot be seen under a microscope unless they are dyed. So, it was Richard Altman who used and dyed. Okay. And observed these organelles under a microscope and explained that these structures are basic unit of cellular activity. In 1898, Carl Benda coined the term mitochondria for these organelles. Okay, red blood cells or RBCs lack mitochondria, as RBCs uses none of the oxygen which they transport, and they do not possess mitochondria. Instead, they derive energy from the different chemical process called glycolysis. Semi-autonomous nature of mitochondria. Cell organelles that contain their own DNA and replicate independently of the nucleus are said to be semi-autonomous. So mitochondria have their own DNA which is capable of replicating independently. Mitochondria are also partly independent or semi-autonomous, as they can manufacture some of the proteins required for their effective functions with the help of their DNA. RNAs, enzymes and ribosomes and often other from the cytoplasm formed under the control of nuclear DNA. The organelles also possesses their own ribosomes called microribosomes. The evolutionary theory, that is the endosymbiotic theory of mitochondria. It is thought that life arose on Earth around 4 billion years ago. The endosymbiotic theory states that some of the organelles in today's eukaryotic cells were once prokaryotic microbes. In this theory, the first eukaryotic cells was probably an amoeba-like cell that got nutrients by phagocytosis and contained a nucleus that formed once when a piece of the cytoplasmic membrane pinched off around the chromosomes. Some of these amoeba-like organisms ingested prokaryotic cells that then survived with the organism and developed symbiotic relationships. Mitochondria formed when bacteria capable of aerobic respiration, photosynthetic bacteria, were ingested. They eventually lost their cell wall and much of their DNA because they were not of benefit within the host cell. Mitochondria and chloroplasts cannot grow outside their host cell. So some of the evidence that is based on the following, that is mitochondria and chloroplasts are the mostly prokaryotic microbes. Chloroplasts are the same size as the prokaryotic cells, okay, divide by binary fission and like bacteria have feeds like proteins at their division plane. The mitochondria are the same size as the prokaryotic cells. Okay, mitochondria also have the same size as the prokaryotic cells. Okay, divided by binary fission. It is also the mitochondria and chloroplast. They both divide by binary fission. And the mitochondria of some protists have homologs at their division plane. Then the next one is mitochondria and chloroplast have their own DNA that is circular and not linear. Mitochondria and chloroplast have their own ribosomes that have 30s and 50s subunits, not 40s and 60s. Okay. Several more primitive eukaryotic microbes such as Giardia and Triomonas have a nuclear membrane but no mitochondria.